Hey, welcome to the second installment of this set of notes. If you missed the first set, we talked about the cell cycle, the life cycle of one single cell, where a cell starts off as a little tiny little baby cell and it grows into a bigger cell. Growth phase, one. And then we go into what we call S phase, synthesis phase, where we make more DNA. Just like in this GIF right here, we see that the one strand of DNA splits into two. We learned this last chapter, and then more pieces are added here and down here, and we end up with two strands of DNA. And then that cell, now with two strands, there's a little bit more growth, needs a little bigger, more protein synthesis like last chapter, and then reproduction. We call M phase, mitosis, meiosis, whatever, where one cell, this piece of clay here will represent the cell, will split into two cells. And then those two baby cells that were just made are going to start all over again. They're going to go through their growth phase and get a little bit bigger. And DNA synthesis again, where one strand of DNA becomes two strands of DNA. And then the cells grow a little bit more. And then M phase again, they both go through reproduction, where we now have four brand new cells. And they're going to go through it again, over and over and over again. We're going to look kind of like this. This is the real culture of bacteria, by the way, and we call this binary fission. When prokaryotes, which are very, very simple, they have almost nothing on the inside. So their insides can very easily duplicate, go to other sides, and the two sides become two distinct cells. You see that happening? One cell becomes two cells. Now we are going to do something very, very similar, except we are not made out of these prokaryotic cells. We are made of more complex eukaryotic cells, where we have all this stuff going on the inside. We have our double helix DNA splitting into two different strands of DNA, two identical strands that we are going to have to separate. And remember, our DNA gets separated out into chunks we call chromosomes. And our chromosomes, notice here that they are together. We have the one side and the other side, the one side and the other side, the two copies, still right next to each other. And each chromosome is lined up down the middle of the cell. For us, we are going to have to very carefully separate them out and pull them across from the middle to the other ends, where each end is going to become its own cell. And just like I've drawn out right here. Get closer. We have made one cell right here full of its individual chromosomes. I only drew 4 because I didn't have room for 46 of them. Go with it. All right. But that nucleus is in the way all around here. So that nucleus needs to disappear. And then these are going to line up down the middle because of chemical reactions. Don't worry about it. And we're going to get these little strings called spindles. That's a vocab word. And those spindles are going to grab each chromosome and tug of war, rip them apart. And then the cell is going to start splitting down the middle. And each bunch of chromosomes, that's what they're called, these individual pieces are now called the chromosomes. Each one gets its own nucleus, and the cell splits right in between those nuclei, and two cells. All of this took place right here. I take it back. This last step, took place in this little section we call cytokinesis. Cytokinesis of the cell. Let's jump into the actual notes now, shall we? This is Unit 6, Cell Growth and Reproduction, Mitosis. Mitosis is the separation of those chromosomes, and then cytokinesis was the cell splitting. That's the easy part. So what all of this is asexual reproduction. How your cells make brand new cells, but without having sex. That's right, one cell became two cells. There was only one parent. We call that asexual. We learned in chapter 1. It is going to give entirely its own A's, T's, C's, and G's, its own DNA, as we saw happening right here. The first cell end up making two strands of DNA, and each baby cell, we call them daughter cells, each daughter cell got one of these DNA. One daughter cell got this one daughter cell got this one. And we're going to use this process to make what we call somatic cells. I believe this was also a vocab word, non-reproductive cells. Right? Reproductive cells we'll talk about later. That's a different set of notes. We're talking sperm and eggs, how to make your baby making cells, right? We're not talking about them. We're talking about every other cell in your body, liver cells, skin cells, brain cells, blood cells. These are all called somatic cells, non-reproductive. And they are going to make identical cells every time. So that's what happens. This is what happens when one cell splits into two cells, like I had on that whiteboard. Starts as one cell and ends up as two new daughter cells. I already said that before, I'm saying it again. They are called daughter cells. I don't know why they're daughter cells and not son cells or child cells. I'm not going to get into that conversation now. Daughter cells. And we need to talk about the stuff on the inside, the DNA. That's the important part. Mitosis is all about the DNA. The rest of the cells, that's easy. That's basically binary fission. Cytokinesis, the cell splits in half. But this DNA, oh no, that's the hard part. That's the part we got to concentrate on. That's what mitosis is all about. We need to get this DNA organized, more organized, and then split. So we need to talk about what the DNA consists of. And it starts off at its smallest level called chromatin. DNA and the proteins floating around the nucleus. This is actually not visible. We can't see this at all. Yeah, I got some pictures of real cells. These are the cells of an onion. These are plant cells, but their nuclei are the same as our nuclei. So take a look, zoom in here, and what we see is each thing here is one cell, and we see a nucleus. One cell, one nucleus. That darker one's a nucleolus. What you don't see in here is DNA. No, what you actually see is a cloud of DNA. You can see the DNA, but we can't see any details. It's just a cloud. So we need to do something about that mess of a cloud and organize it. In this picture, it's this mess in the background. That's the loose DNA. This DNA right here and here, it's here floating around. And your cells are going to organize them by braiding them like hair, twisting them all up, wrapping them all up, and you are going to get what we call a chromatid. One of these, this is actually one chromosome. We'll get there, but it is made of two sides. And one side is a chromatid, the other side is the other chromatid, two identical chromatids. All of this chromatin became one chromatid, all of this chromatin became the other chromatid. And that's what we have here. One side, the other side. And they're held together by a protein called a centromere, which I don't have here. I'm going to, okay. Future me is going to edit that in where you have a little bit of protein dot, maybe it has drawn as a dot right here called a centromere, holding those two chromatids together. And it's going to, that's what's going to help keep us organized. And this whole structure, the two chromatids held together, is known as a chromosome. Two sister chromatids held together. And how many chromosomes do we have again? 46, right? 46 strands of DNA. One, technically, one strand of DNA cut into 46 pieces. That means we have 46 of these X shapes floating around. You got 23 from your mom, 23 from your dad, so you have 46. So how are we going to do that? How are we going to organize all this? Well, we got to start from the beginning. So this is where we start off, a cell living its life. This is one cell here. This is the nucleus. And these are organized, we haven't talked about yet, called centrioles. They're going to be part of mitosis. We need them. So we are not in that part yet, though. This is what we call interphase. Right here on the cell cycle, we are talking about here, here, and here. This whole section of the cell cycle. We're not here yet. G1, S, and G2. The growth phase, the cell is just going to get bigger. And then we move on to the S phase, where one strand of DNA splits into two. Bet you're getting sick of this picture. And then we move on up to G2 phase, where it will grow a little bit more, and these centrioles are going to get replicated. through protein synthesis from last chapter. And now we are able to move into the M phase, mitosis. Mitosis is in different sections. We call them phases. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. And that last step was cytokinesis. We will get there next. Now, you're not writing this page yet. This is more of a helpful hint. This is how I remember things. Prophase prepares the cell. Notice that that cloud of DNA, the chromatin, gets much more organized into the chromosomes, and the nucleus holding it disappears. We are now prepared. Prophase, prepared. And the next phase is metaphase, or middle. M for middle. The DNA lines up down the middle of the cell. All these chromosomes line up in the middle. Chemical reactions make that happen. And then we have anaphase, where DNA is split apart. A for anaphase, A for apart. So they all get these things we call spindles attached, these lines, these strings, and they rip them apart. So anaphase, apart. And then last is telophase, not telophase, telophase, where we're going to make brand new nuclei. Each pile after anaphase of DNA needs to be kept nice and safe in a nucleus. So telophase kind of sounds like telescope. I think telescopic. I think eyes. So it looks like some weird cartoony eyes to me. That's how I remember it. And then the last step is not on the list on our page yet, but cytokinesis is when the cell splits in two. That is not part of mitosis. Mitosis stops here, and then cytokinesis splits. So a few keywords to help us understand things going into this. First one, centrioles. I already said that. These are these structures. They're going to make those spindle fibers. Those things right here, they are going to spread away from each other, releasing the spindle fibers behind them. These spindles, these strings, are what's going to grab the chromosomes. They fan out and separate the chromosomes. So let's go into the order end. This time, much more animated version of it. Prophase, remember, prepares. Before, we could not see any DNA in here. Right? It's a cloud of chromatin, and our eyes, even with a microscope, can't see it. But in prophase, the chromatin condensed into chromosomes. And no, I did not draw 46 of them. There should be 46 of them. I ran out of room. Just go with it. Chromosomes become visible. And then the nuclear envelope, the membrane around the nucleus, dissolves away. Meaning the chromosomes are now free to float around the cell. These centrioles, they are going to stop that from happening. So they're going to move to opposite sides, leaving those spindle fibers behind them, which will be, again, used in metaphase. As they're moving, they're also releasing a chemical that your chromosomes don't like. So they're trying really hard to get away from this guy with the chemicals, but they don't want to get closer to this guy with the chemicals either. So they are going to line up down the center. We call this metaphase, middle, right down the center of the cell. And in this process, notice here, the spindles attached to the chromosomes. Before they weren't. Look, see, a gap, a space. Hold on to that thought. That'll come in handy later on. But right now, if all things work the way they should, these gaps will close. The spindles will attach to the chromosomes. That's metaphase. So recap, prophase, chromosomes showed up. Metaphase, they line up in the middle, and the spindle's attached. And now the actual moment of mitosis doing something. The chromosomes are going to split apart. The centrioles are going to pull. They're going to tug of war. They're going to rip them, and they get ripped. Now we got two identical sister chromatids. Now they're their own personal chromosome, split apart, and they're going to be pulled to opposite sides of the cell. Recap: prophase, chromosomes show up. Metaphase, they line up down the middle. Anaphase, ripped apart and pulled to opposite sides of the cell. And then telophase. Now that we got our own piles of DNA, identical piles of DNA, mind you, see, black and black, green and green, orange and orange, yellow and yellow, they are the same things. We're going to keep them separated by giving them their own nuclei. Now they're nice and protected and safe. And then now that they're safe, they don't need to be so organized. They're going to break back down to that cloud of chromatin, and we're not going to be able to see them again, not until the next time they do mitosis. And that's mitosis in a nutshell. But we're not done yet, because the whole point was to make two cells, and we still got one cell here. Two nuclei, two strands of DNA, but only one cell. We need to finish this. Cytokinesis. And this is the simplest part, really, cytokinesis. Like I said, not technically part of mitosis, but we are going to finish the job. And it does come in two different categories. They're going to behave differently between animals and plants. Here you see the animal version of it, where one cell starts pinching inward in the middle. You're looking at step by step by step by step by step by step here, right? And then eventually pops into two cells. That's cytokinesis. Cyto means cytoplasm, the stuff inside the cell. Kinesis means to move. Cytoplasm, moving. Cytokinesis. In animal cells, like I said, what you witnessed here, we are going to get a pinching in motion. There's a term for this. It's called a cleavage furrow. The word cleave means to cut, which is why there are knives called cleavers. It is going to cut, it's going to fold inward and break this cell into two cells, splitting it right in half. That is the way animal cells do it. Plant cells, on the other hand, are going to be a little different. See, there's a problem. They have a cell wall, and cell walls are not going to bend inward like this. Cell walls are made out of wood. I should say wood is made out of cell walls, but you get the idea. They're not going to bend very easy. So instead, they are going to form something in the center called a cell plate. A cell plate forms between the two nuclei. So here we got a cartoony version and a real version, and this is the real version that comes from the onion slide that I showed you, where we have our cell with our chromatin floating around as a cloud. Prophase, the chromosomes become visible. Metaphase, they line up down the middle. This way it's sideways line, not up and down, but works the same. Anaphase, they split apart. And telophase, they get their own nuclei, but look right here in between. I can't zoom in, so you're going to have to bring this closer to your face. There is a wall, a miniature wall forming. We call that a plate. That's how plants do it. Where animals are going to bend inward from the outside in, plants are going to grow from the inside out, start in the middle and work their way to the ends. That cell plate here and here will eventually become a brand new cell wall, and we will have two distinct individual cells. Two smaller cells, but two cells. And then they will grow into full-size cells and the plant will grow. Or for animals, the two halves will be small cells and they'll grow and you will grow. And that is it for mitosis, for the how it happens. But I got one question for you. I got to ask, why is this important? Why do you care? What is mitosis doing for you? Well, one, like I just said, it allows you to grow. In order to grow, you need more cells. A bigger body has more cells, plain and simple. And very important, it allows you to repair yourself. This kid got cut. He's bleeding because he lost skin. So now he needs to replace that skin. He needs to make new skin cells. And one last thing before we leave, I have a question. I showed you what happens when things work correctly, but what happens if things get messed up? What happens if the chromatids fail to separate? Remember, I asked you to hold on to this thought, where we have the spindles not attached to the chromosomes during metaphase, and then they do attach. What happens when they don't attach? What if that accident happens? And yes, that accident does happen. If the spindle fibers do not attach, you see right here, I only got 3 chromosomes so I can make them bigger and you can see it. The spindle here is not attached to this yellow one. It's so close, but not close enough. So in anaphase, when they pull, the whole chromosome moves over. This guy has nothing to pull. Whole thing goes over here. So now we're going to have a cell that has too many chromosomes over here and not enough chromosomes over here. And we have two very different cells. Too much information, not enough information. And that can mess you up because that can cause a mutation. In fact, that is a mutation. Luckily, we are talking about mitosis. We're talking about making new skin cells or something, right? Your immune system will recognize this and fix it. It will destroy these cells and you'll start again and you'll be fine. The problem comes in the next set of notes. What happens if your immune system's not involved? When we make our sex cells, your immune system's not involved. Gentlemen, if you make sperm, ladies, if you make eggs, those are the things that will be used to make your kids. If that doesn't get fixed, that's a big problem. And we will talk about that next time. And for now, thanks for being here. Good luck. I will see you in class.